Good afternoon, everybody. I would like to thank all of you for staying this late. My name is Benedict Anchang. I'm a postdoc at Stanford and working in Dr. Plevretis' lab. Understanding cancer heterogeneity has been a key focus in several recent studies. In particular, tumor cells have been shown to exist in phenotypically and functionally distinct states. During this presentation, I would like to demonstrate how we can isolate these cell states from a mixture of cell states in an unbiased and less subjective manner, and I will show an application in breast cancer. Our study is motivated by two recent studies. The first study by Gupta and colleagues in their Cell paper demonstrated evidence of phenotypic switching between cancer cell states. In this study, they isolated stem-like and basal-like and luminal-like cells from a parent breast cancer cells, and over time, these sorted cells showed characteristics of the parent population. Using a simple Markov model, they were able to show evidence of phenotypic switching between cancer cell states. The second study by Zaperi and Porter gave an alternative explanation for the same phenomena. They argue that starting with a contaminated cell population could still result in stem-like and non-stem-like cells. So this means that understanding cell state transitions will require a strong evidence of pure cell state population. In practice, biologists get their data sequentially in two dimensions, conditioning on two markers using qualitative thresholds such as high, low, positive, negative, or mid. For example, in the Cell paper, the gated stem-like and luminal-like cells from an EpCAM positive population. The EpCAM negative population was characterized as basal-like cells, while the stem-like populations were characterized as CD44 high, CD24 negative, and EpCAM low. The luminal-like cells were characterized as CD44 low, CD44 high, and EpCAM high. The problem or the question with such a characterization is that how do you quantitatively define high, negative, low when you are not looking at all the markers at once? This slide shows you how getting the same data with two different schemes can result in two different populations. Scheme A gets the data first on marker 1, then on marker 2, and you could see clearly two populations. However, scheme B gets the data instead first on marker 2, and then on several partitions on marker 1, you could see more distinct populations. So this means that to successfully or manually get the data, you have to use the right order and the right partitions of the markers. This brings us to the goal of our study, which is to provide the biologist with a simple, automated, unbiased gating strategy for his or her data. Our approach uses clustering, which is to identify the cells that are in the same cell state, and a decision tree which formalizes the manual gating process. I will start with the first step. We use a non-parametric mixture model to estimate the cell states. This approach takes into account the uncertainty about which cell state a cell belongs. So if we assume that our cell measurements are actually independent and coming from a mixture of unknown functions, then the density of an individual cell can be written as the sum of weighted product unknown densities. The key is to come up with the posterior distribution of the cell states. And we make use of an EM-like algorithm, which was introduced by Benedict Anchang and colleagues in the Statistical Journal paper in 2009. This approach uses a kernel and a bandwidth, and it's deterministic. And the posterior modes from the posterior distributions are used to assign the cell states. Note that maximizing such a posterior distribution could result in the wrong assignment of boundary cells. And since silhouette measures have been shown or used as a diagnostic tool to evaluate the performance of clustering algorithm, we reassign all the cells with negative silhouettes to their most likely neighbors. We iterate this process until the overall silhouette score remains unchanged. This step optimizes the cell states. We now turn to the next stage of the algorithm, which is to come up with a decision tree corresponding to the gating scheme. We make use of recursive partitioning techniques in particular the technique by Horton and colleagues in the Statistical Journal paper in 2006. In this step, we test the global null hypothesis of independence between all the markers and the nominal response cell states. And we select the marker with the strongest association based on adjusted p-values. We then implement a binary split, and we select the marker, and then we recursively repeat steps 1 and steps 2. This approach actually overcomes the marker selection bias and the subjective manual gating process. And the C3 function in the R package party has a flexible stopping criteria. The output for such an algorithm is a decision tree with the root node corresponding to the marker that separates the data best, and the leaf nodes correspond to the marginal distribution of the cell states. We then applied our algorithm on one of the cell lines in the cell paper, that is the SOM15 breast cancer cells. Using our algorithm, we're able to isolate 5 cell states of populations, mainly based on CD24 and EPCAM, denoted here as P1, P5, P3, P2, P4, and P5. If you compare our gating strategy with that in the paper, you will see that they started off with EPCAM, and then they gated their 3 populations, stem-like, luminal-like, and basal-like from using CD24. Our approach starts with CD24, and using several cutoffs from EPCAM, we could identify these 5 populations. Comparing our populations with their populations and summarizing the results in a cross-classification table as shown, we observed that the basal-like population actually show evidence of more cell states. And there seems to be a mixture of stem and luminal-like populations, which also show evidence of contamination after sorting. We then took our gating algorithm and asked one of our lab members to see if she can reproduce the populations in on the machine. And this panel shows the the gating scheme on the training data, and this shows the gating scheme on the test data. So she was able to reproduce all the cell populations. And this really validates experimentally our gating strategy. We are now in the process of really looking at what is happening with these 5 populations. And I would just like to show you some preliminary results. Looking at the 5 populations after sorting, we see that at 2 hours, this is the progression of the cells, and at 2 weeks, this is the progression of the cells. And it means that after some time, the cells seem to generate the same cellular phenotype in vitro. Now, if we zoom into one of the subpopulations, in this case, subpopulation 2, we realize an interesting pattern. We see that immediately you sort the data, it seems the population 2 transition quickly into all the cell states, as shown in this, by these heat maps. The rows are the cells, and the columns are the markers. And after 2 weeks, we see a similar pattern with the same population. This is interesting in that it seems that once you sort out the cells, they want to stay together. And with this interesting observation, I would like to conclude. So in this study, we developed and applied an automated gating algorithm to identify and get out cell states that are less likely to be contaminated with mixtures of other cell states. Also, our model can easily be scaled to a large number of markers. And we observed evidence of phenotypic switching between 5 cancer cell states instead of 3 cell states by the cell paper. And these cell states will really warrant further characterization, including even gene expression profiling. I would like to take this opportunity to acknowledge all those who took part in this study. Special thanks goes to Sylvia, Dr. Privitis, who is our lab leader. I would also like to thank Mary Doe for really generating this clean data, and Zizao and Zira for their fruitful discussions, and other lab members. I would like to thank NCI for funding the project, and I would like to take this opportunity to thank RECOMB and all the collaborators or organizers for giving me the opportunity to present my work. Thank you for your attention.
Questions for Benedict? Yeah.
I have a very naive question. So if you identify the population starting from the clustering with all the markers together, I mean, by clustering that space, what do you need the gating for after? I mean,
Pardon?
why do you need the gating? I mean, if you already identify the population using the clustering in the space defined by all the markers, why do you need the gating?
The clustering gives you, you mean the population of the clustering, and why do you need the...
Yeah, I mean, the clustering gives you the population assignment for each cell, right?
Yes.
So why do
the
you need
practice, in practice, as I said, you have to sort these populations with in the machine.
Okay, it's for the... Okay.
So it's for, so that's why I said in practice you don't, no matter, with all the gating algorithms that are out there, it's really difficult to translate it into a practical, so no matter how you come up with an optimal algorithm, you still have to give the biologists a simple gating scheme for the data. Yeah.